Welcome back to the course Fundamentals of General Immunology. So today we are going to start our module 3, whose title is Antigenic Information and Immune Specificity, Antigen Processing and Immune Communication. So in this module, we will try to understand the determinants of antigenicity, why a protein can be immunogenic in nature, what are the factors responsible for that, what are immunoglobulins, what are antibodies, how this diversity in antibody chains can decide the specificity of an antibody against a particular antigen. That we will try to understand in this module. So the lecture number is 8 and the title is Epitope Architecture and Determinants of Immunogenicity. So let's try to understand the immune complex related structure of a protein. So we will define this term called epitope. Now epitope, as we already looked into our earlier lectures about what are antigens. So antigens are any foreign substances which are entering into the body and raising an immune response. Here we will define the term epitope. And epitope is also the name of epitope, we also called it antigenic determinants. So these are defining the antigenicity of a protein. These are very specific structures. These are located on an antigen. So these are very specifically located on an antigen, and this antigen could be a protein, could be any other substances which can coming out from outside. This could be from virus or bacteria, and they are recognized by the immune system, particularly the antibodies or the B cell or the T cell. So let's try to look into this structure of antigen here, which is a protein structure. So these are a chain of amino acids. So these are in the form of a protein, and we can look into either these could be an N terminal or this could be a C terminal, and we can see that there are multiple folds of the proteins available. So for example, the foldings are here, the foldings are here and here. Similarly, these areas are also exposed, these areas are also exposed, this is also exposed. So these structures which are exposed form of a protein is what we call epitopes. Now generally these epitopes are the structures which are present on the antigen and can be identified by these antibodies. So we will look into this antibody a little later in this lecture only, where you could look into that these specific domains which are present. Now these are also proteins. So these are protein-protein interactions actually. So these are complex protein, and the domain of the protein is also interacting to this particular site of an antigen. So this particular site of an antigen is what we call epitope of an antigen. So there are criteria which decides this antigenicity, or what are the factors which could cause this particular area called as an antigen, which we'll look into in details. So let's try to see a few more details of these epitope structures. Now as I said that epitopes are proteins, and once the proteins are formed from the ribosome in a cytoplasm. So for example here, if you see that the proteins are formed from the messenger RNA in the cytoplasm with the help of this ribosome, the proteins are formed. Now once these proteins are formed, these proteins are either decorated by the structures which are called glycosylation. So let's try to understand a few details of epitopes. There could be two terms which we need to understand. One is called conformational and the other is called linear. Now once the proteins are formed into the cytoplasm after the ribosome bound to messenger RNA, these proteins are converted into different forms, and these forms are acquired through the post-translational modification. This could be a glycosylation, This could be a phosphorylation or any other post-translational modification. Now, the conformational aspect of the protein are very important, because once the proteins are dissolved in different pH or temperature or say buffer, a lot of things which are there where the protein is dissolved is deciding the conformation of a protein. Say if the pH is acidic, lower pH, the protein conformation may be different. If the pH is high, say alkaline, the conformation of the protein might be different. If the temperature, for example, at 37, the temperature, ambient temperature, the protein might fold differently. If there is a lower temperature, protein might fold differently. Say for 37 degrees centigrade, you can see a different conformation, while at 4 degrees centigrade, you see different conformation. Similarly, in buffer, say there are normal buffers such as PBS, phosphate buffer saline, or say we have tris buffer where we add some kind of an agent to kind of support or stabilize the protein molecules. Now, these buffer may fold different protein differently. Now, because of this reason, the conformation of a protein is changed. Conformation of the protein is changing gradually based on the structure, which are based on the pH, temperature, and buffer. So, here we can see that this protein actually conformationally differently folded, and therefore, the area which are exposed can be identified by the antibody differently. And therefore, we could say that the epitope formed by amino acid brought together by protein folding is what we call conformational epitopes. While in linear epitopes, the structures are actually free from the conformational aspect. So, these are linear chain of amino acids. So, they are not changing. So, they are not conformational. They are not buried or say exposed conformationally. They are just exposed linearly. So, they are not say deviated by the change in temperature, pH, or buffer. In that case, we could see that these are linear epitopes. So, the epitopes formed by a continuous sequence of adjacent amino acids is what defined as linear epitopes. So, we will see gradually that how these linear and conformational epitopes, how these are useful to raise an immune response and how these are different. So, the conformational determinants, linear determinants, and new antigen determinants, these are the three terms again we have to understand in context of epitopic structures. Now, once the protein is formed, so essentially these proteins are produced, and once if by chance we are doing a denaturation means using any agent that can strengthen the protein. Say the protein folding can be changed. In that way, the denaturation can lose the epitopic determinant. So, once there is a denaturation, then the epitopic structures are changed, and that is what the conformation is very important for a protein. While the linear determinant, which we have discussed in the last slide, even there is a denaturation, the linear structures are intact. So, the structures which are present on the linear epitopes are always identified by the immunoglobulins or the antibodies intactly as compared to the conformational epitopes. While in conformational epitopes, we can see that the epitopic structures are completely destroyed. While in linear epitopes, even though denaturation happens, the structures are intact. There could be third possibility, which we call neoantigen, and neoantigens are something very unique. And what could be the possibility to produce a neoantigen is, suppose we have a protein and by chance the epitopic structure are buried inside somewhere. So, there could be a possibility that protein folded in such a way that antigenic regions are just buried inside. They are not exposed outside. So, the body may not identify those as an epitope, although these are antigen, but these are they are buried inside. And therefore, once there is a proteolysis happening, or say destruction of the protein by the host cell proteases. So, there are many proteases which are present in the host cell that could decide the destruction of protein in a different form. So once there is a proteolysis or proteolysis, the protein gets multiple fragmentation, and out of those fragments, some could be acting as an antigen, determinant, or epitope. Now this could happen because the buried antigens are now exposed, the epitopic regions are now exposed, which could be identified by antibody as a new one. And since these are exposed, these are produced post proteolysis, these are new, these are not already present in the protein, these are in the buried form, therefore these are called neoantigens, or new antigens, which are created because of the proteolysis event in the cellular thing. So there could be a possibility, multiple possibility, where we could see that there could be a conformation epitopes, there could be a linear epitope, or there could be a neo-epitopes which are produced post proteolytic event. So let's try to understand this again, a new concept which is called the naive and immune individual. Now the naive individuals are the individuals or the human beings which are not exposed to any antigens in past. So if there is no exposure to the antigen, there is no immune response against those antigens. That means if there is a person who is infected with some kind of an pathogen coming in contact with this naive individual which are not exposed to these pathogens earlier, what will happen that these infected patient will actively transmit the pathogen either by the aerosol route through the air, circulation of air, or through direct contact, or through by any means. There could be a possibility that these antigen can be transferred to this naive individual, and once these are transferred to the naive individual, since there is no immune response, the person may start suffering from a disease, and then we could see that the symptoms of disease are coming into that individual which are exposed to the antigen from an infected individual, and those individuals are now symptomatic, so they can start showing signs and symptoms of a disease. While in other case, suppose this person is already exposed to these antigens in the past, and therefore these individuals are having some sort of memory in the form of a plasma cell which can produce an immunoglobulins that can bind to these antigens very categorically, and therefore if they are coming in contact with this infected individual, even though there is a spread of the pathogen to these individual, body recognize this as an already encountered pathogen, and they can raise an immune response to destroy these antigen because they are previously exposed to those antigens, and therefore you don't see any symptoms in these individuals. So this is simply a vaccine biology which will deal in our later classes, but just for the time being, antigenicity is such an important thing that can decide the disease condition or health of an individual. So a perfect homeostasis between the antigenicity and the immune response is important in a human body. So let's try to see that what determine the immunogenicity. What determine the immunogenicity in terms of the epitopic structure. So as we know that the immunogenicity is the ability to induce a specific immune response, and it's influenced by the character of an antigen. Now see, there are different nature of antigen which we need to understand here. The first one is foreignness. The foreignness stands for how distant the antigen is from a human being. For example, if there is an antigen coming from Homo sapiens to Homo sapien, we are very close to, the antigens are very close evolutionary, and therefore you see there is an immune response, of course, because this is non-self. So suppose there is one human being getting some kind of an organ transplant or anything to individual number 2. So for example, this is 1 and this is 2. You could see the immune response against this transferred antigen, but this immune response will be subtle as compared to once the person 2 will get something from, say, a different species altogether, not an Homo sapien. For example, other species, for example, a pig or a mice. These are distant than human, and therefore you see the foreignness can define by how far the evolutionary distance a person is having. And therefore, we can say that the foreignness is what decides the immunogenicity or the nature of antigen. More the foreignness, higher the immune response. That is what the baseline. The second is molecular size. Now, we understand that the proteins are of varying size. They have a varying charges also. Some proteins are smaller in nature, some proteins are larger in nature. As a rule, the larger molecules are better immunogen as compared to the smaller one. And we have seen the example of heptanes, which are very small molecules, unable to raise an immune response unless and until they are carried through some larger carrier proteins. So, when they are mixed with the larger carrier protein, we could see the immune response. But in general, the smaller immunogens, say for example 1000 Dalton, critically, if there are any proteins which are of size 1000 or lower in size, may not be that efficient immunogen. So therefore, we can see that the size is very critical. So more the bigger size of the protein, more conformational we can look into, and therefore we could expect the larger immune response in the human body. The third one is chemical complexity. So of course, proteins are complex in nature, which we have discussed, even though protein complexity in terms of amino acid is critical. So proteins are made up of different amino acids. Some are basic in nature, some are say hydrophilic in nature, some are aromatic in nature. In general, the aromatic residue, more aromatic residue in a protein can cause little higher immunogenicity as compared to the protein which are having low aromatic amino acid profile. And also when we are talking about some of the proteins or some of the molecules which are polymeric in nature. So if there is a repetition of the polymer, a similar kind of polymer, if it's repeated, then probably we may not get a very good immune response. But if it is a polymer which are having a varying concentration or varying substitution in the protein, then we could see that these polymers could be highly immunogenic in nature. Physical form of a protein could decide, particularly when we say that there are two forms physically. Either the antigens could be in the form of a particulate form or the soluble form. So the particulate antigens are mostly more immunogenic in nature, while the soluble, the one which is kind of a liquid in nature, may not raise an immune response to that extent. Again, the denatured protein are generally more immunogenic than the native protein, because they are exposing more of the epitopic structure post denaturation, and therefore we can see that once the proteins are denatured, more of the epitopic structure are exposed, and the body immune response can divert towards that more efficiently. Therefore, more particulate form of antigens are more immunogenic, while the soluble form is little less immunogenic in nature. I'll just quote an example here. We all know cellulose. So cellulose is a polymer of glucose molecules. Cellulose is largely non-immunogenic in nature unless until we see some variation of cellulose. So therefore, these cellulose are radially used in tissue engineering or tissue material, because the body recognizes these structure as to some extent self, and therefore you don't see an immune response happening, and therefore the cellulose build up. So the material which build up because of the cellulose or cellulosic material may not raise an immune response to that extent as compared to the non-cellulosic material. And therefore, we need to understand that the materials which are used for tissue engineering are mostly defined or directed towards the way if they are non-immunogenic in nature. So these are some of the examples where we see that foreignness, molecular size, chemical complexity, and the physical form of the protein can decide the immunogenicity. Next one is again another important factors which are related to the host. Particularly, these are not related to the protein, these are related to the host. Some of them are the genotype. For example, say the genetic factors or the molecules which can identifying those epitopic structures, such as major histocompatibility complexes, which are in short called MHC, which we'll see later in detail. How these are expressed, whether these are having a high affinity towards the epitope, whether these are low affinity towards the epitope, can decide the immunogenicity of an individual. So therefore, the genetic makeup of an individual, say how robust the immune response is there in that individual, can decide the immunogenicity. So there are multiple determinants of immunogenicity, and one such thing which people are thinking of is how the host factor is responsible, how the gene regulation can be responsible for immunogenicity. Second is immune status. Now this is again very important, because age, for example. So we always say that the young ones and the elders are kind of not showing the immunity in a proper sense. Why? Because the young individuals are not developed their immune system in a proper way. Say their primary and secondary lymphoid organs are not developed in a proper way, while in case of elder individual, the things already got exhausted, or the proper organs which are there to support the immune response in the human individuals are not functioning in a proper way because of the old age, and therefore age is a very important factor. Just quoting an example here of a polio virus. So the polio virus is mostly infecting to the young ones, the age group of 7 to 8 years, or the young individuals, because the receptor of the virus, for example, suppose this is a polio virus, these viruses can carry some kind of an epitopic structure on the surface. They can be identified by a specific receptor on the host cell. So this is a host cell, this is a virus. Now these proteins are expressed only at a particular time. So these receptors which are there in the host can express only at a particular age time, and therefore once there is a receptor, they can identify this virus structure as an epitope and raise an immune response, or say viruses could be causing some sort of infection to those host individual. While in the elder age group, or the more 40, 50 years of age, these receptors are not at all expressed, and therefore there is no chance of poliovirus to do infection in later time point of life cycle. So therefore age is very critical. This is just an example. Nutrition is also play a very important role. Say what you eat will decide the immune response of an individual, how proteinous diet a person is taking, how different kind of food supplement in terms of say vitamins or minerals or carbohydrate, how these nutrition status of an individual is there will decide the immune status of that particular person. Particularly sometimes there is an immune compromised individual in case of some infections such as HIV, which is a viral disease called human immunodeficiency virus that specifically targets the immune cells, and therefore we always see that those individuals suffering from HIV might have a lower immune response, or immunocompromised individuals. These are also genetic factors which are responsible for that, which is beyond the scope of this course. So we will just try to understand that there could be an immune factors or gene factors which are responsible for immunocompromised individual. Prior exposure is also important. Say if the person is already exposed to an antigen, the pre-existing immunity will protect the individual. That's what we discussed in the earlier slides, where we discuss about naive individuals where the person is not exposed to a pathogen, while a person who is already exposed might have a memory immune response in the form of a plasma cell which can protect the individual when there is a future exposure to the same pathogen. So these are the three factors which are actually related to the host site. Next comes the four other aspects, such as administration or dose, how these antigens are given. For example, what is the route of antigen inoculation in the human body? For example, if the antigens are inoculated by subcutaneous route, it produces a very strong immune response. followed by intradermal, intramuscular and intravenous, because these are very sustained. So once the antigens are deposited into the subcutaneous layer, we called it subcut, the antigens may stay there for a longer duration of time. Once the antigen is staying longer duration of time in the subcutaneous layer, the immune cells are recognizing this antigen very efficiently, and therefore you see a very high immune response in subcutaneous area when we are using the antigen to inoculate there. And therefore once we are raising an antibody for research purpose, we generally use this particular area called subcut in order to raise an immune response. While intradermal and intramuscular is again a fast route, because the antigen may clear very fast from those areas. While intravenous is very, very fast, because immediately once anything is going into blood circulation, it immediately pass on to the next level, and therefore this is the fastest route, and therefore the immune response is least in those cases. Second is dose. Of course, the antigen, when they are inoculated into or the antigen entering inside the host body, the amount of antigen could decide the immunogenicity. If the amount is very high, that's also not good. If the amount is very low, that is also not good. So when we are raising an immune response or antibody against an antigen, we could look into the optimal dose of an antigen, because too little antigen may not raise an immune response, and if there is a more antigen, then we could lead to a condition called immune tolerance, because the body immune system is showing exhaustion because of high amount of antigen. Similarly, in the human condition, the disease may happen based on the concentration of antigen. When the person is infected with a bacteria or virus, the amount of antigen entered inside the body can decide the disease progression, the incubation period, what we call, say the entry of the pathogen till the person starts showing sign and symptom. That time is called incubation period. This depends upon the amount of antigen that entered inside the body. If the amount is very high, we could see that the incubation period is very low. When the amount is very less, the incubation period may be very high. So doses are important. Of course, the interval, duration, say once you, so this is particular term useful when we are raising an antibody for some research purpose or antibody for prophylactic purpose. When we are using an antibody to treat against a disease, we have to raise those antibody in some other animal. Now in that case, time interval is important. Say once we are inoculating, say this is an immunoglobulin response, and if this is time, if we draw a graph, and suppose if we are putting a first immunogen here, and if we give a time of say 14 to 21 days, and then we are raising when we are inoculating another antigen here, then we could see a larger immune response in that case. If we are not giving this window of 14 to 21 days, and when we inoculate some antigen here itself in the middle of the cycle, we see that there could be a higher immune response, but the immune response may not be that high, and therefore this interval is important. So we have to give some proper interval when we are raising an immune response in terms of second dose of hand. We call it booster also sometimes. So this is a first exposure and this is a booster. So the time interval is important. Third is adjuvant, and of course we have looked into some heptane like molecule which are not immunogenic in nature unless until you add some carrier. Here these adjuvants are the agent which are added with the vaccine to enhance the immune response. So, so for example, suppose this is, we will take the example of the earlier subcutaneous. So suppose this antigen is inoculated subcutaneously to an area of a human body. This will stay longer and raise an immune response. But suppose if I add a molecule here along with this immunogen, so that this molecule can attract the macrophages or some kind of other antigen presenting cell towards the site, so that this antigen can be recognized in a better way, and therefore this substance which are forcing the other immune cell to come to the site and effectively raise an immune response is what we call adjuvant. These are mostly related to vaccine biology, which we will see later, that these are the agents which are added on the vaccine, because this kind of phenomena is important when we are looking into to give protection to the individual by an immune response. All right. So we'll look into some other details in our next class about how these antibodies are defined, how this antibody can be differentiated into different types, subclasses of antibodies, and how these are very important in terms of binding to the antigen. Thank you.